Good evening, students. Previously, we explained the chapters and bits of simple tissues, complex tissues, special tissues. Now, we will explain anatomy in plants and secondary growth. Anatomy in plants, we have 5 concepts: TS of dicot root, TS of monocot root, TS of dicot stem, TS of monocot stem, TS of dicot leaf and TS of monocot leaf. Dicot leaf is also called dorsiventral leaf. Monocot leaf is also called isobilateral leaf. So, we discuss anatomy of TS of dicot root, anatomy of TS of monocot root. The basic difference, even though both are roots, there are some basic differences in dicot root and monocot root. The best difference is in dicot root, pericycle produces lateral roots and cork cambium, whereas in monocot roots, pericycle produces only lateral roots, it does not process cork cambium. In dicot root, the xylem is tetrarch and exarch maximum, whereas in monocot root, the xylem is polyarch and exarch. In dicot roots, central medulla is reduced due to the extensive growth of metaxylem vessels. In monocot roots, a central medulla is prominently present. These are some basic differences. Then dicot stem, monocot stem. Dicot stem shows U-stele, limited number of vascular bundles arranged in the form of a ring. Epidermis shows trichomes. Hypodermis is collenchymatous. General cortex helps in conduction and storage. Endodermis is also called star sheath. Pericycle is sclerenchymatous. Vascular bundles are conjoint, collateral, open. Whereas in between two vascular bundles, medullary rays, and at the center a prominent medulla will be present. Coming to monocot stem, it shows epidermis, sclerenchymatous hypodermis, parenchymatous ground tissue, and numerous vascular bundles irregularly scattered in the cytoplasm or ground tissue, which is popularly called atactostele. Atactostele is supposed to be the most advanced character in almost all monocots. Every vascular bundle is conjoint, collateral, and closed. Open in dicot means cambium is present in between xylem and phloem. Closed in monocots means cambium is absent in between xylem and phloem. Then second, in monocot stem, general cortex, endodermis, pericycle, medulla, medullary rays are completely absent. Coming to the, and in monocot stem, just below the protoxylem vessels, there is a special structure formed by the disintegration of cells called lysogenous cavity or protoxylem lacuna, which is absent in dicot stem. Then leaves, upper epidermis, lower epidermis, adaxial, abaxial surfaces. Particularly in a monocot leaf, upper epidermis, some of these cells become bulliform cells, large cells with hygroscopic in nature, helping in rolling and unrolling of leaves. They are called bulliform cells. In the monocot leaf and dicot leaf difference, mesophyll is divided into palisade tissue and spongy tissue in the case of dicot leaf. Mesophyll is homogeneous chlorenchymatous tissue in the case of monocot leaf. Regarding vascular bundle, both are same. Endodermis, not I don't say endodermis, it is bundle sheath, but because of the presence of Casparian thickenings, it is regarded as endodermis. Vascular bundles in both the leaf, dicot and monocot leaves, are conjoint, collateral, and closed. Cambium is completely absent. That means leaves absolutely not show any secondary growth. Now, we discuss some important bits in anatomy. First, this is a matching tabular form. Match the following and choose the correct combination. Troponatans, Pisum sativum, Ricinus communis, Nicotiana, tobacco. We have to remember one tabular form. Monarch and exarch. In dicot root it will come Tropa. Diarch and exarch, Nicotiana. Triarch and exarch, Gossypium. I don't say Gossypium, it is Pisum. Tetrarch and exarch, it is Gossypium. This triarch and exarch condition is also found in hibiscus. So, Pisum and hibiscus. Pentarch and exarch in Ricinus. Octarch and exarch in a plant called Castanea. These are the variations in a vascular bundle of a dicot root. Variations in the vascular bundle of a dicot root. Tropa, monarch condition. So, when it coming to the combinations, Match the following and choose the correct answer. Trapa is, the answer is 2 here. A5, Trapa nutans monarch condition. Pisum sativum triarch condition 3. Ricinus communis pentarch condition 1. Nicotiana tabacum diarch condition 2. So, A5, B3, C1, D2. So, Trapa natans is monarch and exarch. Pisum sativum is triarch and exarch. Pisum sativum is triarch and exarch. Ricinus communis is pentarch and exarch, and Nicotiana tabacum is diarch and exarch. So, A5, B3, C1, D2. The matching of this is correct is second option. Roots contain radial vascular. This is an assertion reasoning question. Roots contain radial vascular bundles. Radial vascular bundles means, what is a radial vascular bundle means? If you take a ground plan like this, xylem and phloem appear in separate radii but perfectly alternate with each other like this. This is called radial vascular bundle. This is xylem, this is phloem, and this is conjunctive parenchyma. Xylem, metaxylem towards the center, or metaxylem towards this medulla, protoxylem towards the pericycle or periphery. This is called exarch. So, this is radial vascular bundle where xylem and phloem are arranged in separate radii. Xylem and phloem are separate radii is radial vascular bundle. The assertion is correct. Cambium is absent in the vascular bundle of roots. Always in the roots, vascular bundles are closed. That means cambium is absent. But because of the absence of cambium, roots are not radial vascular bundles. So, assertion is correct, reason is correct, but reason is the not correct explanation of assertion. So, answer both A and R are true. R is not the correct explanation of A. Another assertion. In a mature root, exodermis gives protection. In a mature root, exodermis gives protection. What is exodermis? The parenchymatous cortex that is present just below the epidermis in the older roots get the deposition of suberin and becomes exodermis. So, what is the chemical present in exodermis? Suberin. Suberin is a poisonous substance. Whenever this comes and deposits, the cells become dead. So, in a mature root, exodermis gives protection is correct. Then, reason, in mature root, epidermis is removed. So, in mature root, sometimes epidermis is removed. Then the protection is given by exodermis. So, exodermis gives protection. In mature root, epidermis is removed. Both are linked with each other. So, A and R are true. R is the correct explanation of A. So, exodermis shows the chemical substance suberin. Suberized cells are present. Continuous collenchyma has hypodermis and discontinuous sclerenchyma in the pericycle are found in the stem of. You have to remember two concepts. Dicot stem in hibiscus and dicot stem in cucurbits. Dicot stem. In hibiscus, hypodermis is present as a, hypodermis is collenchymatous. So, collenchymatous hypodermis is present as a continuous ring. Sclerenchymatous pericycle. Sclerenchymatous pericycle is present as patches. Exactly opposite in cucurbits. Collenchymatous hypodermis present as patches. Sclerenchymatous pericycle present as a continuous ring. So, remember these two basic differences. In both dicot stems of hibiscus and cucurbits, collenchymatous hypodermis is present as a continuous ring and sclerenchymatous pericycle is present as patches above each vascular bundle. In the case of cucurbits, collenchymatous hypodermis is present as patches, collenchymatous hypodermis is present as patches, sclerenchymatous pericycle is present as a continuous ring. Here the question is, continuous collenchymatous hypodermis and discontinuous sclerenchymatous pericycle are found in the stem of Helianthus. So it is Helianthus. Cucurbita, Aristolochia, Oryza are just to confuse, but Cucurbita is opposite. Continuous collenchymatous hypodermis will not be there. Patches of collenchymatous hypodermis will be there, and pericycle is present as a continuous ring. So for this the answer is Helianthus, sunflower. The shape and arrangement of vascular bundles in a monocot stem are. Whenever you see the ground plan of monocot stem, it appears like this. It shows epidermis without trichomes. Just below the epidermis, there is a sclerenchymatous hypodermis. So in dicot stem, hypodermis is collenchymatous. In monocot stem, hypodermis is sclerenchymatous hypodermis. The total inner portion is filled with parenchymatous ground tissue. Total. So in perfect and general cortex, endodermis, pericycle, medulla, medullary razor apse. In the vascular bundle, in the ground tissue, numerous vascular bundles are irregularly scattered. The peripheral vascular bundles are large in size. The central vascular bundles are small in size. So numerous vascular bundles scattered is popularly called atactostele. So atactostele is a special modification or what you call advanced characters in monocot stem. So the shape and arrangement of vascular bundles in the monocot stem are not top shaped and circular ring. Top shaped and circular ring is dicot stem. Not oval and circular ring. They are not at all arranged in circular ring. Top shaped and scattered is also wrong. Answer is oval shaped and scattered vascular bundles throughout the ground tissue. Second option. Protoxylem lacuna in a monocot stem is. Protoxylem lacuna is formed by the disintegration of cells, not by the expansion of intercellular spaces. So protoxylem lacuna or lysogenous cavity in a monocot stem present just below the protoxylem vessels formed by the disintegration of cells. Hence, it is not a schizogenous cavity, it is not a schizolysogenous cavity, it is not a mucilaginous cavity, it is basically a lysogenous cavity which is formed by the breakdown of cells. So, answer is 2. There is no cambium between the vascular tissues of. There is no cambium between the vascular tissues of. There is no cambium between vascular tissues of dicot root, monocot root, dicot stem, monocot stem. So for this answer is dicot root, and monocot stem also. Here the answer is not 1, it is 4. 1, 2, 4 only. It is not 1, it is 4. There is no cambium between the vascular tissues of dicot root, monocot stem. Cambium is present in the vascular tissues of dicot stem. So answer is 4. Based on the number of xylem strands, arrange the following plants in ascending order. Arrange the following plants in ascending order. Arrange the following in ascending order means ricinus 5 strands, pentarch and exarch. Gossypium 4 strands. Nicotiana 2 and Trappa 1. So ascending order means 1, 2, 4, 5. That means 4, 3, 2, 1. Trappa is 1, Nicotiana is 2, Gossypium is 3, Ricinus is 5. So the ascending order is lesser is Trappa 1, next is Nicotiana 2, next is Gossypium 4, and last is Ricinus 5. So answer is 4, 3, 2, 1. This is an assertion reason. Passage cells of the endodermis are present opposite to the protoxylem elements. Passage cells of the endodermis are present opposite to the protoxylem elements. Passage cells have thin walls without Casparian bands. Passage cells of the endodermis present opposite to the protoxylem elements is correct. Because they do not have Casparian bands and because they possess thin walls, they are not present opposite. Because of the absence of Casparian bands, they are called passage cells. So the answer here is both A and R are correct. R is not the correct explanation of A. Passage cells have thin walls without Casparian bands is correct. Passage cells of the endodermis are present opposite to the protoxylem vessel elements is correct, but they are not correct explanation of A. So, answer is both A and R are correct. R is not the correct explanation of A. In a primary dicot root, suberized cells are. Suberized cell means the cells which are surrounded by suberin. Just now I told exodermis present just below the endodermis in a old root is suberized. Endodermis, the cortical layer present around the stele. Endodermis is popularly called cortical layer present around the stele. This endodermis on some cells, radial and tangential walls. On some cells, radial and tangential walls show the depositions of lignin and suberin. Those ligno-suberized bands are called Casparian bands. So, endodermis shows lignin and suberin, and those ligno-suberized bands on their radial and tangential walls are called ligno-suberized bands. So, according to this question, in a primary dicot root, the suberized cells are present. One is exodermis, the other is endodermis. Both contain suberized cells. Pericycle is a dead tissue and conjunctive tissue is a parenchyma tissue which not show suberin. So, suberin depositions are found in exodermis and endodermis. Assertion. Exodermis of a dicot root prevents the leakage of water from cortex. Exodermis of the dicot root prevents the leakage of water from cortex. Exodermis is suberized cells. It not only gives mechanical support but also prevents the excessive loss of water. In the absence of epidermis, exodermis helps in the absorption of water. In the absence of epidermis, exodermis helps in the absorption of water is wrong. So, exodermis of dicot root prevents the leakage of water from cortex is correct. In the absence of epidermis, exodermis is helping in the absorption of water is wrong. So, the answer is A is correct but R is false. Third option will be the answer for this assertion and reasoning question. Another assertion reasoning questions. The root hair are multicellular outgrowths of rhizodermis. Rhizodermis, epiblema, piliferous layer are the names given to a root with root hair. So, root epidermis, some of the epidermal cells gives rise to root hair. Any cell that gives rise to root hair in the epidermal cell is called trichoblast. This is also important. So, epidermal cell which is having the capacity of producing root hair is trichoblast. Epidermis, the root hair are multicellular outgrowths on rhizodermis. Root hair are tubular extensions of epidermis is correct. But root hair are not multicellular, they are unicellular. Here the assertion is given as root hair are multicellular outgrowths of rhizodermis. Rhizodermis is correct but they are not multicellular outgrowths. They are unicellular hair-like structures. Root hairs are the tubular extensions of epidermis is correct. So, here A is false, R is true. A is false, it is not multicellular, they are unicellular in nature. Number of passage cells in the exodermis of Zeamays. What are passage cells? The endodermal cells without suberin depositions are passage cell. Without suberin depositions are passage cell. These passage cells are present opposite to protoxylem. Definitely present opposite to protoxylem. So, because of the opposite to protoxylem, one passage cells in present in Trapa, 2 passage cells are found in Nicotiana, 3 passage cells are found in Pisum, 4 passage cells are found in Gossypium, 5 passage cells are found in Ricinus, 8 passage cells are found in Castanea. Zeamays is a monocot. Zeamays is a monocot, that means it shows polyarch and exarch xylem. So, whenever polyarch and exarch xylem is there, opposite to protoxylem vessels also numerous passage cells are present. So, number of passage cells in the endodermis of Zeamays is many, not 4, not 8, not 0. The answer is many passage cells are present in the endodermis of Zeamays. Why? Because Zeamays is polyarch and exarch. Anatomically, a monocot stem differs from dicot stem in having conjoint collateral vascular bundle is a common feature in monocot stem and dicot stem. Phloem parenchyma is absent in monocot stem, present in dicot stem. More xylem vessels in each vascular bundle is the perfect answer for this question. Anatomically, a monocot stem differs from dicot stem in having more xylem vessels in each vascular bundle. Why? Because polyarch, I don't say polyarch, it is atactostele. Numerous vascular bundles are irregularly scattered in the ground tissue. Answer is 3. The structures absent in monocot stem are phloem parenchyma is absent in monocot stem. Protoxylem lacuna or lysogenous cavity is present in monocot stem. So, second option will not come. Endodermis and pericycle are absent in monocot stem. They are present in dicot stem but absent in monocot stem. Medullary rays are absent in monocot stem. They are present in dicot stem. So, answer is 3. 1, 3, 4. So, the structures that are absent in a monocot stem are phloem parenchyma, endodermis, pericycle and medullary rays. Protoxylem lacuna is present in monocot stem. This is another assertion reasoning. In a dicot stem, eustele is present. In a dicot stem, eustele is present and eustele means the ground plan, cortex is less, stele is more. Cortex is less, stele is more. And some of the epidermal cells gives rise to trichomes. Eustele means limited number of vascular bundles arranged in the form of a ring. This is called eustele. Cambium is present in between xylem and phloem. Xylem towards the lower side, phloem towards the upper side. And this is medulla. And these two are medullary. This zone between two vascular bundles is medullary ray. So, this is eustele. Eustele means limited number of vascular bundles arranged in the form of a ring. So, in a dicot stem, eustele is present is correct. In a dicot stem, stele is bigger than cortex. In a dicot stem, stele is bigger than cortex is correct. But because the stele is bigger than cortex, eustele is not present. Eustele is present right from the day one as the plant grown on the face of the earth. So, both A and R are correct. R does not explains A. In a dicot stem, eustele is present is correct. Limited number of vascular bundles arranged in the form of a ring. In a dicot stem, stele is bigger than cortex is wrong. It is correct but not related to assertion. So, A and R are correct. R is not the correct explanation of A. Anatomically, monocot root differs from dicot root in having. That means the basic difference between monocot root and dicot root. Protoxylem, polyarch xylem, correct. Well developed medulla, correct. Pericycle gives rise to adventitious roots. Pericycle does not give rise to adventitious root either in dicot root or in monocot root. So, anatomically monocot root differs from dicot root in having is, one is polyarch xylem, numerous xylem vessels alternating with phloem, radial vascular bundles, well developed medulla. In a monocot root at the center a permanent prominent parenchymatous medulla is present, whereas in a dicot root that medulla is very much reduced or crushed due to the extensive growth of metaxylem vessel. So, answer for this is 1 and 2. Polyarch xylem and well developed medulla are found in monocot root. Consider the following statement and match with the dicot stem. Vascular bundles are arranged in a ring around the pith. Correct. Vascular bundles are radial, wrong. Vascular bundles are radial is wrong. They are conjoint, collateral, wedge shaped collateral, conjoint, endarch and open type of vascular bundles is correct. Wedge shape, conjoint, collateral, endarch and open type of vascular bundle is correct. Continuous ring of hypodermis with living mechanical tissue is correct. Continuous ring of hypodermis with living mechanical tissue means collenchyma. So, in some plants it is present as a continuous ring of hypodermis with living mechanical tissue. So, answer for this is 1, 3, 4. Second is not wrong because vascular bundles are not radial, exarch and closed. Radial, exarch, closed, all the three are not at all related to a dicot stem. So, 1, 3, 4 comes under the answer. In which of the following characters a monocot root differs from a dicot root? What is the basic difference between monocot root and dicot root? Radial vascular bundles, closed vascular bundles, everything is same. Trichoblasts are present in the epidermis in both large pith. If a large medulla is present, that is monocot root. If the medulla is crushed or absent or reduced, it is dicot root. So, answer is 2. In a dicot stem, the sequences of tissues in centrifugal direction. Centrifugal direction means from base to apex. So, first epidermis, it is centripetal from top to bottom, then hypodermis, then general cortex, then endodermis, then pericycle, then phloem, then cambium, then xylem, last medulla. So, this is from top to bottom, centripetal. Centrifugal means like this, from center to periphery. Answer is medulla, xylem. So, medulla, xylem, cambium, phloem, pericycle and hypodermis. So, this is called centrifugal explanation and this is called centripetal explanation. From periphery to center is centripetal, from center to periphery is centrifugal. So, if it is asked in the centripetal manner, epidermis, hypodermis, general cortex, endodermis, pericycle, phloem, cambium, xylem, medulla. Suppose the same question asked in centrifugal manner, medulla, xylem, cambium, phloem, pericycle, endodermis, general cortex, hypodermis, epidermis. So, the answer for this question, arrange or identify the regions of a root from centrifugal manner means in a dicot stem, the sequence of tissues in a centrifugal direction is pith or medulla, xylem, cambium, phloem, pericycle, hypodermis. In a isobilateral leaf that of a grass, bulliform cells are present in the both epidermal layers is wrong. Bulliform cells are present only in the upper epidermal cells. Mesophyll is differentiated into palisade spongy tissue is wrong, because mesophyll is divided into palisade and spongy tissue only in a dicot leaf, not in a monocot leaf like grass. Mesophyll is not differentiated into palisade and spongy tissue. It is correct. Mesophyll is absolutely not divided into palisade and spongy tissue. Mesophyll is filled with homogeneous chlorenchymatous tissue. Veins form reticulum. Veins form reticulum is also wrong, but some grasses like smilax show reticulate venation. So, correct answer for this question is mesophyll is not differentiated into palisade and spongy tissues. Third option. So, study the following table and identify the correct match. Collateral conjoint closed vascular bundles, collateral conjoint endarc closed vascular bundles, radial separate polyarc vascular bundles and closed, collateral conjoint endarc and open vascular bundles. For this answer is for A5. Collateral conjoint and vascular bundles are found in monocot leaf. B4. Collateral conjoint endarc closed vascular bundles are found in endarc and closed vascular bundle means monocot stem. Radial separate polyarc vascular bundles are found in polyarc is monocot root. So, second option. Collateral conjoint endarc and open vascular bundles are found in dicot stem. So, collateral conjoint closed vascular bundles in leaf. Collateral conjoint endarc closed vascular bundles in monocot stem. Radial separate polyarc closed vascular bundles in monocot root. Collateral conjoint endarc and open vascular bundles in dicot stem. So, A5, B4, C2, D1. Answer 4 for this 23rd question. The outstanding character by which a root differs from a stem. That means how a root differs from a stem in its anatomical features. By seeing anatomy, how can you differentiate between root and stem? Prominent endodermis with Casparian thickenings is correct. Prominent endodermis with Casparian thickenings is correct. Xylem is always exarch is correct, because in stem it is endarc, in root it is exarch. Then presence of epiblema. Presence of epiblema cannot be a differences between dicot, root or stem. Presence of simple and homogeneous cortex is correct. So, answer is 1, 2, 4. The outstanding character by which a root differs from a stem is prominent endodermis with Casparian thickenings, Xylem is always exarch. Presence of more simple and homogeneous cortex. So 1, 2, 4 are correct. The outstanding character by which a root differs from a stem. In a transverse section of a typical dorsiventral leaf, that is dicot leaf, phloem in the midrib is situated. Phloem in the midrib means phloem in the midrib region, not facing adaxial because upper epidermis. Outside the border parenchyma, wrong. Facial, both adaxial and abaxial is wrong. Facing abaxial epidermis is correct. Abaxial means lower epidermis. In the vascular bundle of any leaf, xylem is present towards the upper epidermis, adaxial surface. Phloem is present towards the lower surface, that is lower epidermis, abaxial surface. So answer is facing abaxial epidermis. Find out the correct statement. Polyarch condition is found in all monocots. Starch sheath of dicot stem is pericycle with Casparian bands. The trichomes of dicot stem promotes the entry of pathogenic microorganisms. Trichoblasts of root epidermis sometimes gives rise to lateral roots also. Starch sheath of dicot stem is the pericycle with Casparian bands is wrong. Why? Because starch sheath of dicot stem is the endodermis with Casparian bands. Endodermis, not pericycle. The trichomes of dicot stem promotes the entry of pathogenic microorganisms is wrong. The trichomes of epidermis always prevents the entry of microorganisms or pathogenic organisms. They don't allow any organisms to go into the root. They stop the entry of any organisms into the root. Trichoblasts of root epidermis sometimes gives rise to lateral roots also. Very, very wrong. Trichoplasts are special epidermal cells in a dicot root or monocot root that gives rise to unicellular green or unicellular special delicate hair-like structure called root hair. They do not give rise to lateral roots, they give only root hair. So answer for this is polyarch condition of xylem or polyarch condition is found in all monocot root. So find the correct statement means polyarch condition is found in all roots. Starch sheath of dicot stem is the pericycle with Casparian bands is wrong. The trichomes of dicot stem promoting the entry of pathogenic organisms is wrong. Trichoblasts of root epidermis sometimes give rise to lateral roots is wrong. Lateral roots are not produced from trichoblasts, they are formed from pericycle in a root endogenously. This is an assertion reasoning question. Monocot stem possess closed vascular bundle. Monocot stems possess closed vascular. What is a closed vascular bundle? Cambium is absent in between xylem and phloem. So every vascular bundle of monocot stem resembles like this: oval, conjoint, collateral, closed. They show two large metaxylem vessels. They show two small protoxylem vessels and a big protoxylem lacuna. Towards the metaxylem vessels you will find phloem. This is metaxylem vessels, this is protoxylem vessels, and this is the cavity formed by the disintegration of some of the cells of protoxylem called protoxylem lacuna or lysogenous cavity. So here, because of the presence of conjunctive parenchyma, because of the presence of parenchymatous tissue between xylem and phloem, the monocot stem possesses closed vascular bundle is wrong. The reason must be in a monocot stem the vascular bundles does not possess cambium must be the reason. So monocot stem possesses closed vascular bundles is correct. Conjunctive tissue is present in monocot stem is correct, but they are not related to each other. So both are correct, but R is not the correct explanation of A. Bundle sheath is absent in. Bundle sheath is absent in. Definitely it is present in vascular bundle of monocot stem made up of xylem fibers, hence it is also called fibrovascular bundle. It is present in the vascular bundles of monocot leaf, it is present in the vascular bundles of dicot leaf. Why? Because in vascular bundles of dicot leaf and monocot leaf they are surrounded by bundle sheath cells. Then what is the answer? Answer is vascular bundles of dicot stem. In dicot stem, vascular bundle are not surrounded by bundle sheath, they are surrounded by single layered endodermis called as starch sheath and sclerenchymatous pericycle. Undifferentiated ground tissue is met within, or undifferentiated ground tissue is found in. Maize root, ground tissue is nothing but medulla. Undifferentiated ground tissue is met with, that means present in. In a maize root, in a Zeamays stem, in a sunflower stem and cucurbita stem. Undifferentiated ground tissue is found in Zea stem. Why? The total inner portion is filled with ground tissue where numerous vascular bundles are irregularly scattered, which is called atactostele. So, answer is 2. Suberin is the important constituent in the cell wall of. Suberin is the important components in the cell wall of. Suberin is a poisonous substance. It is absent in epidermis of root. It is found in exodermis of root. It is found in cork of periderm. Cork deposited with suberin. Cork with suberin becomes resilient, compressive, very light in weight and thermo-insulating properties. Sometimes cork without suberin. Sometimes cork is not deposited with suberin. They are called phalloids. So, cork with suberin is found in exodermis of the stem and cork of periderm. Pericycle of dicot stem is sclerenchymatous. Epidermis of root absolutely does not contain suberin. So, suberin is present in the exodermis of root and cork of periderm. Both definitely shows suberin. Vascular bundles where the phloem without parenchyma are found in. Phloem without parenchyma is phloem parenchyma. Phloem parenchyma is found in maize root, phloem of maize root. It is found in the phloem of helianthus stem. It is found in the helianthus root. It is absent only in maize stem. Why? Monocots are the plants where the phloem absolutely does not possess phloem parenchyma. So, maize stem. First option. How a dicot leaf differs anatomically from a monocot leaf. So, now the question is how a dicot leaf differs anatomically. Presence of parallel venation. Dicot leaf does not possess parallel venation except a few like Calophyllum erynchium. Presence of stomata on both sides. In a dicot leaf, the condition is called hypostomatous. This is in dicot leaf. What is hypostomatous? Stomata are restricted only to lower epidermis. Monocot leaf. Monocot leaf is called amphistomatous. Monocot leaf is called amphistomatous. That means stomata are equally present in both upper and lower epidermis. Both upper and lower epidermis. So, hypostomatous condition is found in dicot leaf. Amphistomatous condition is found in monocot leaf. So, how a dicot leaf differs anatomically from a monocot leaf is not by presence of parallel venation, not by the presence of stomata on both sides, not by the presence of only spongy parenchyma, only by the presence of palisade and spongy parenchyma. Palisade tissue are long columnar cells present towards the upper epidermis. Spongy is loosely arranged cells on the lower epidermis. So, by the presence of palisade and spongy parenchyma, a dicot leaf differs anatomically from a monocot leaf. It is an assertion reasoning question. In a monocot leaf, entire mesophyll carries out both assimilation and exchange of gases equally on both sides. It is correct because it is homogeneous. In a monocot leaf, stomatal frequency is same on both sides and mesophyll is homogeneous. So, monocot leaf entire mesophyll carries out both assimilation and exchange of gases equally on both sides. Why? One is presence of amphistomatous condition that is given. In a monocot leaf, stomatal frequency is same on both sides. And another is total mesophyll is homogeneous. So, for this answer is 1. A and R are correct. R is the correct explanation of A. Another assertion reasoning. Stele of monocot stem is referred as atactostele. Correct. Stele of monocot stem is referred as atactostele. In monocotyledonous stem, scattered conjoint collateral closed fibrovascular bundles are present. Because of the presence of scattered conjoint, collateral, closed fibrovascular bundles only. The stele of monocot stem is called atactostele. So, for this also the answer is A and R are correct. R is the correct explanation of A, first option. Coming to the another assertion reasoning. Secondary growth is normally absent in monocotyledonous plants. Secondary growth is normally absent in monocotyledonous plants. In monocots, the vascular bundle lacks fusiform initials. Secondary growth is normally absent in monocotyledonous plant is correct. Reason: in monocots, the vascular cambium lacks fusiform initials. In monocots, no chance of formation of vascular cambium. If vascular cambium itself is not formed, how come it produces fusiform initial? So, reason is totally wrong. A is correct, R is false. Third option. Identify the incorrect statement from the following. In many monocot leaves, the bundle sheath extensions are made up of both sclerenchyma and chlorenchyma. In the leaves of grasses, the bundle sheath cells are homologous to endodermal cells. This is correct because they contain Casparian thickenings. In many monocot leaves, biseriate bundle sheaths are present. Biseriate, definitely 2 layers. In bundle sheath is uniseriate and maize leaf. This is also correct. So, wrong is, in many monocot leaves, the bundle sheath extensions are made up of both sclerenchyma and chlorenchyma is wrong. It is made up of sclerenchyma and parenchyma. In a dicot leaf, bundle sheath extensions running from endodermis or bundle sheaths to upper and lower epidermis is parenchymatous. Whereas in monocot leaf, the bundle sheath extensions are sclerenchymatous. So, correct answer is in many monocots, it is the incorrect statement in these 4. The number of xylem strand in the second plant is 5 times more than that of the first plant. Whereas the number of xylem strands in the third plant is 4 times less than that of xylem strands of fourth plant. Identify the ascending order of the 4 plants according to the number of xylem strands in the stele. Trapanutens 1, Nicotiana 2, Ricinus 5, Castanea 8. So, answer is 1. Answer is 1. Trapanutens, just to confuse you, the number of xylem strands in the second plant is 5 times more than that of the first plant is given. Okay. So, answer is 1. Now, this is a 3-column matching. Structure of plant, tissue, organ of the plant. Trichoblast, epidermis, root. Correct. Trichoblast in the epidermis gives rise to root. Passage cells, endodermis, root. Correct. Epiblemma, epidermis, stem. Wrong. Epiblemma is the other name of epidermis with root hair. It is not present in stem, it is present in roots. Trichomes, epidermis, monocot root. Monocot root shows only root hair. Monocot roots does not give root hair. So, trichomes, epidermis, monocot root is wrong. So, correct is 1 and 2.